Okay, our next speaker is Susie's colleague, Johnathan Puddick, also from the Cawthron Institute. And call on Johnathan to come forward. And his talk is titled Blooming Buddies, Understanding Microcystin Sequestration in Non-Toxic Cyanobacteria and its Ecological Significance.
All right. Thanks for having us back in the country. So I was here last 2 years for what was then the National Cyanobacteria Workshop, and thanks for changing the name as well, because it made it really easy to jump over the ditch and come back to Australia. And I just sort of love this sort of tight-knit community that we've got going on here, and who knows, in sort of another few conferences' time, it might be the Pacific Cyanobacteria Workshop if we keep getting people from all over the place. So today I'm going to talk to you about a research project which we recently began, which has been funded by the Marsden Fund of New Zealand. And so this funds fundamental research from a wide range of different sciences. And what I love most about coming to cyanobacteria conferences is that I don't really need to talk to this slide here, because Sandra's already told us about the diversity of cyanobacteria. We've learned all about how they're taking a lot of interest now, because we're sort of creating environments where they love to dominate and they're blooming. And we've learned a lot about cyanotoxins and how that's caused human and animal health problems. And so there's a lot of work going into cyanobacteria around the world. But what I do want to tell you a little bit about is my favourite cyanotoxin, microcystins. And so this is what I've spent a lot of my research career working on. And so these are potent hepatotoxins, so they accumulate in the liver and they affect protein phosphatases and basically mess around with the cell regulation there. But also they've been shown to promote liver tumours, so chronic low-level doses are a big problem, and so this is why I think that such a big concern is because it's not just those acute effects, it's the chronic effects over a long period of time, and I've spent a lot of time working on developing more sensitive analytical techniques to be able to look at these. So not all cyanobacteria produce microcystins, but a large array of cyanobacterial genera do. And so the strains which are capable of producing this toxin, they have a gene cluster called microcystin synthase, which codes for a complex of enzymes which basically put together all these amino acids into this cyclic ring structure like we see down here. And so the holy grail of a toxin researcher, a cyanotoxin researcher, is really trying to figure out the ecological function, the whole why do cyanobacteria put so much energy in actually producing these compounds. And we know it's not due to predation, because the genes which encode for it developed a lot longer before there was a long time before there was actually anything to predate on them. And so I like this structure, this graphic here from Nealon et al., which puts together a lot of the sort of different functions which have been posed for microcystins at least. And the reason I like this is because it sort of speaks to there possibly being dual functions for microcystins, and that makes it really difficult to understand. Now, where I did want to get to with these introductory slides, though, was to the point where I got to sort of say that as a cyanotoxin research community, we have got to a sort of place where it's sort of an us and them, thinking about the toxic strains which are able to produce toxins and get a competitive advantage for it, and the non-toxic strains. And we're starting to make assumptions where we just disregard that non-toxic community and start to think about the toxic community. And we've had to do this to move forward, and I've done it myself. But a couple of years ago, I came across this paper by Phelan and Downing, a South African duo. And what their study did was they provided exogenous microcystin to a non-toxic strain of Seneca cystis. And so that microcystin accumulated in that cyanobacteria, and about 50% of it ended up being localised in the thylakoid membrane. And what they saw was that this didn't affect the viability of that cyanobacteria, but it did reduce photosystem 2 activity. So it was having a clear effect on the non-toxic cyanobacteria. And what it made me think about was this paradigm where we sort of think about those toxic-producing cyanobacteria producing toxin which has some sort of competitive advantage which we don't really completely understand, and keeping it to themselves. And I started to think, well, you know, what if that's not actually what it's like? What if these toxic cyanobacteria, what if they produce this toxin, but they don't keep all of it to themselves? What if they exude some of that? And what if these non-toxic cyanobacteria, which are forming part of their community, are taking it up and actually using it for themselves? So they're getting the competitive advantage as well. And what we're seeing is actually cooperation between these two populations where all of them are able to thrive. And so this is what led me down this wonderfully murky path which the Marsden Fund decided to actually fund and to look into this research question a little bit more. And so I separated that out into 3 key objectives. So I wanted to know what is the actual diversity of cyanobacteria which are able to sequester microcystin. So it's been shown in Seneca cystis, but what about a range of different cyanobacteria? But just taking up that microcystin doesn't actually show that you're getting a competitive advantage from it. So we also wanted to know, if you're taking up that microcystin, does that actually give you some advantage as if you weren't being supplied it? And the last question was, does this actually occur in the natural environment? So is it actually relevant to us, or is it just some weird sort of phenomenon that we're seeing in a laboratory and, you know, we can disregard it? So, off onto objective number 1. And so the way that we set up, we thought this is going to be super simple, and it wasn't quite as simple. And I'll maybe tell you a little bit about that in the question times if we've got some at the end. But what we wanted to do was to take a range of non-toxic cyanobacteria from our culture collection at Cawthron, and we wanted to provide them with microcystin. And then we'll grow them for a couple of days, and we'll harvest those cyanobacteria onto a GFC filter. The exogenous microcystin, which is left behind in the media, will flow through. We'll wash out that filter with some fresh media. We'll extract that and we'll analyse for microcystins. And so if we don't detect any microcystins like over here, that means that microcystin's still in the exogenous out in the media, and we're not seeing sequestration. And if we do detect microcystins, then it's been taken up by the cyanobacteria, and that's why we've been able to detect it. So what we end up finding was in the 17 strains that we've assessed so far, the majority of those did actually take up microcystins. And so one of the nice results that we've seen is that a Seneca cystis species from New Zealand was able to uptake microcystins. So that was nice that Phelan and Downing's work was actually repeatable here when we tried it again. Another nice aspect that we were able to see was that 2 nostoc species, one which grows in planktonic form, so free-floating as colonies, and one which grows as a benthic form as mats, we were able to see microcystin uptake in both of these. So two species of the same genera giving the same response. Some of the results which weren't exactly what we were expecting though was the results that we see here for Microcystis wesenbergi. For a species of cyanobacteria which is so closely related to toxin-producing Microcystis, we were expecting to see microcystin uptake there, but we weren't seeing it. And something that we're going to have to grapple with over the next little while as more results come to life and we delve a little bit further into this project is why are these genera of cyanobacteria not accumulating microcystin when these other ones are? So we don't really understand what the difference is between cyanobacteria which will sequester microcystins and ones which won't. So we also thought that whilst we're doing this experiment anyway, we might as well have a little bit of a preliminary look to see if we're seeing any competitive advantages. So this was only a 2-day culturing experiment, but we had a look at the cell levels that we saw. And so then the green here is just the starting concentration, and then over the period of 2 days, we hopefully see some growth, but some are slower growing and we see none. And so there's no real strong pattern there to say that adding microcystin to these cultures is increasing their growth. But at the same time, I might not expect that as well. So there's been studies out there which have had toxic strains of cyanobacteria and non-toxic strains of cyanobacteria sort of in growth experiments. And what they find is that the toxic strains have competitive advantages under some environmental conditions, but not under all environmental conditions. And that is why we wanted to do Objective 2 a little bit differently from this. And so we've got these nice photobioreactor systems which we developed in-house at Cawthron. And so they're multi-vessel. We've got 6 vessel, and we can control the light intensity, the spectral quality, the pH, and the nutrient supply. So it gives us a lot of manipulation over the environmental conditions that these cyanobacteria are going to grow in. And with those 6 vessel, it means we can do these lovely central composite design experiments relatively quickly. And so we can get these growth optimum curves where we can put any sort of attribute on this y-axis over here. Maybe it's growth rate, maybe it's photosynthetic efficiency determined by PAM fluorometry. And we can figure out what are the optimum growth environmental conditions to get maximum yield of whatever our parameter is. And so what we're imagining that we're going to find is that when we do these experiments using microcystin supplementation, is that we're going to see this growth optimum probably coming up in a different place from when we'll see it with just the control strain, not supplemented with microcystins. So for Objective 3, what we want to do is know about this in the natural environment. And so what we're planning to do with that is to use these dual fluorescent probes. And so we're going to use a fluorescent probe for the microcystin molecule itself, and a fluorescence in situ hybridization probe for the microcystin synthase genes. And so with that, we'll be able to figure out which cyanobacteria in our samples are actually able to produce the microcystin, and where our microcystin is actually being localized in our samples. And what we're hopefully going to see is that in the natural environment where we've got the toxin producers and the non-toxic cyanobacteria coexisting together, is maybe it's actually present in the other samples. And so whilst we're just going to start with grab samples being sent in from lakes around New Zealand, what we hope to do is to be able to adapt these probes to use with some sampling techniques which I presented a couple of years ago. And the video curse is maybe going to stay on, but basically with these probes, they're metal, and what we do is we tip liquid nitrogen into them. And so if in about 20 seconds, what it does is it freezes the water on the sides of the probe, and we're able to take it out. And we end up getting these vertical profiles like this, where the cyanobacteria are embedded in ice sheets. And so we get these fine-scale profiles of the cyanobacterial community. And what we've been using that for in the past is to look at scum formation of microcystis, and to look at microcystin production when these microcystis scums form. And what we've found is that microcystin production is really localised just at that top air-water interface. And so this is sort of at a resolution of millimetres right now. What we hope is that by using these probes with this technique, that we might be able to take that millimetre scale to the micrometre scale and try and link up microcystin production with the transfer that's going on. So just to summarise, because Phil's sitting down there trying to edge me off, so microcystin sequestration, we're able to show that occurred in a range of different cyanobacteria, but at present we haven't really been able to show that any competitive advantage is awarded from that. In the future, what we're hoping is that we can use these cryosampling techniques to be able to understand that on a cellular level and out in the natural environment as well. So just a nice slide to leave you there for question time, for some acknowledgements from our funders and people that have helped us out with this project, and thank you all for listening.
Thanks, Jonathan. Plenty of time for questions. So, just wait for the microphone, please. Yeah, thank you.
Those poorly connected neurons that were in the back of my mind, I looked up in the break. It was Svi Dubinsky who worked on production of exopolysaccharide and then leaking out of cells that were damaged or senescing as a result of photoinhibition. And so that story that you've put up there, that accords at least with my mental model of cells at the surface which may be simply leaking photosynthate in response to a high light environment and then carrying stuff out with them. It'd be interesting to follow that up.
Really interesting research project. I was just wondering if you've done the sort of positive to positive control where you feed microcystins to cyanobacteria that would normally produce them. Because maybe they'll tell you something about whether it's an active or a passive process that's happening.
What a wonderful segue to what's caused me so much hassles with my microcystin sequestration experiments. So my original plan was to do exactly that, because I had sort of a theory, that things which are a lot more closely related are going to be more prone to microcystin sequestration. And so we've got a range of strains which produce a range of different toxin profiles. And so I was going to feed different toxin profiles to toxin-producing strains and then analyse them by LCMS. And that's how I'd be able to identify that we'd actually sequestered the microcystins I'd supplied, rather than, them just changing their production during the experiment. The only problem was when I go to analyse these samples of sequestered microcystins by LCMS, because it's a really specific technique, I can only detect that molecule in its own form. And what I think is happening is that the microcystins are getting bound to proteins and peptides when they get sequestered, when they end up on those sort of membranes. And I can't actually detect them by LCMS. We can detect them by ELISA because it's sort of a little bit different. It's sort of, as long as it's in solution and in your extract, you sort of can get the antibody interacting with it. So yes, it was something that I really hoped to do. And I just haven't been able to, and that's actually why I'm not as far along with this project as I wanted, because I did spend a lot of time sort of figuring out, can I liberate the microcystin? Surely that I can do this, because I'm like a true advocate of LCMS technology. And yeah, so the ELISA crowd has sort of got me over a barrel now.
Jonathan, I've got a question. Back in the 1990s, David Bourne was looking for bacteria which could take up microcystin and metabolise it. And he found that a species of Pseudomonas had microcystin transporters in its membrane. Microcystin is a polar molecule, and therefore it would require a transporter to transport it into the cell. Have you looked for the transporters, the potential transporters in the cell walls of your cyanobacteria?
No, but I sort of think about it, and I think about, the putative microcystin transporter and the microcystin synthase cluster sort of sits in my mind quite a lot. But no, I haven't done any genetic lurking around, but I sort of think that will be one of the things as we move on, that sort of a cunning master's student out there will want to get in on. Don't mention it to my master's students, though, because they've got things to work on already.
Still got time for another question if you'd like, if anybody. No? I think that's it. Okay, well, thank you, Jonathan. And we'll move on to our next presenter.